All right, hello. We're going to start chapter 6, which is photosynthesis. To me, this is probably the most important chapter we're going to look at all semester. Photosynthesis is absolutely necessary. Remember what we've been talking about. What is the source of all energy here on this planet? What is the ultimate source? It is the sun. But sunlight energy cannot be harnessed and used just by absorbing it. Photosynthesis is plants taking the energy of the sun with carbon dioxide and water and making sugar. And so the sugars are going to be the basis of all life on this planet. But sugars are more important than just having that source of energy that we need. Let me tell you about an experiment that was done. I don't know the exact numbers, but here's what happened. A vat of soil, and let's say it's full of dirt. Let's say there's 1,000 pounds of dirt in that vat. Now we take a 1 ounce acorn and plant the 1 ounce acorn in there, and you add water to it, and you know what's going to happen, right? A tree is going to grow. So after 10 years, you have this mighty oak tree that is growing up. Now, after 10 years, you take the oak tree, you cut it down, or you just pull it up by roots and all, and weigh the soil. Now, after 10 years, let's say that the soil weighed 999.5 pounds. Okay, so only 1/2 pound of the soil was missing, and it was probably less than that. I'm just showing you here so you understand this concept. Now, how much did the tree weigh? Now, this is a dry weight, so all the water was removed from it, and the tree weighed 3,000 pounds. So that is 2,000 pounds more than what the soil initially weighed, and we only lost a 1/2 pound of weight from the soil. So where did all this weight come from? I mean, trees weigh a lot. Where does all that weight come from? Because this experiment proved it's not coming from the soil. There's no way it's coming from the soil. Only a 1/2 pound, and we've got 3,000 pounds. Where did all that weight come from? Maybe it was water. Now, remember I told you, this is dry weight. There's no water weight in this. So where did the weight come from? And this is the miracle of photosynthesis. All that weight, 3,000 pounds for that tree, came out of the air. So what plants do when they undergo photosynthesis, they will take the carbon dioxide molecule, they rip the oxygen off, release that, so plants do release oxygen, which we need. And so when we break down sugars, we release carbon dioxide, so we're really working together with each other. But so the plants rip off the carbon dioxide and the oxygen, release the oxygen, and then start putting together these carbon molecules, carbon atoms, to form a larger molecule that now has weight. And as this goes again and again and again, remember, plants will use those sugars for the instant energy, but they will store most of it in the form of either starch. Remember what starch is? Take a glucose molecule and start putting them together to form a big giant molecule of starch, and that's for energy in the future. So when, like today, it's just a miserable day outside, it's rainy, it's windy, the sun's not shining. So when the plant needs energy, it can just start breaking off those sugars off the starch and use it. But remember also, when we talked about the molecules of life, plants will also take those sugars and start arranging them in a lattice work format to form cellulose. Now that is that tough plant material that allows the plant to grow up. And then some plants will produce an enzyme called lignin that will go into the cellulose and form it into woody tissue. So right outside the window here, I have a big giant oak tree. That thing probably weighs well over 2 tons. Where did all that weight came from? It came from carbon dioxide in the air. As carbon is captured, we call that carbon fixation, and we'll talk about that as we go through this chapter. So let's get ready and start going through the PowerPoints here for this chapter. And And we will, let me get our camera adjusted here so that you can see what we're dealing with. All right, and this chapter starts off with a talk about biofuels. What is a biofuel? Let's see if I can get this arranged out just a little bit better here. All right, what's a biofuel? It is a fuel that is made from organic processes. Fossil fuels, which is what we use most of the time for our fuel, is it's made out of plant material from ancient forests. It's a limited resource, so coal and gas. Biofuels are made from organic matter that's not fossilized. It's a renewable resource. So biofuels we can keep making more of, but they're made out of crops such as corn, soybeans, sugarcane. Not so much sugarcane, mainly corn is the main thing that they make ethanol from. You realize that when you go to the gas station, your gas has 10% ethanol in it, and so that comes, most of it, from corn, some soy. They're starting to experiment with some other things, switchgrass and algae to make these biofuels. So coal, petroleum, natural gas are fossil fuels. The remains of ancient forests is a limited resource. Biofuels such as oils, gases, or alcohols are made from organic matter that's living. It's a renewable resource. In the United States, biofuels are produced mainly from food crops. Now, we're going to be talking about here in a minute. You're going to write a paper on this. It's just a short little thing. We'll talk about that in a second. We're turning food into fuel. Only in America would we consider doing something like that. But so we take food and convert it into biofuels. And so here's just a picture of this scientist trying to determine how we can better make biofuels. That's not really going on there, but and here's the switchgrass that they're starting to look at. Now, in this class, we're going to be doing 2 or 3 papers called How Do You Vote? Probably 3, I'm hoping, as we go. I know summer term, it goes really quick, but how do you vote? And so biofuels. Right now, whether you want it or not, 10% of your gasoline is ethanol. So what I want you to do is look for 3 positives about biofuels and 3 negatives. This is going to be really easy. This paper only needs to be just a page long. There's 2.5 paragraphs will do it. Give me 3 advantages, and to do that, just go to the internet, type in advantages of biofuels, find me 3 good advantages of biofuels. And then, perfectly okay, introduce what you're saying and talk about you're going to list advantages, and then you can cut and paste. As long as you give me quotation marks on either end, and how you're going to reference these, as soon as you put the second quotation mark, give me a parentheses and put in it where you found it. So let's say an advantage of biofuel. Biofuels release less carbon dioxide into the air than fossil fuels. And then that's in quotation marks. Then give me a parenthesis. www.biofuels.com, accessed on May 22nd, 2020, and that's all you need to do. Put the other parentheses on there, you're finished. You don't need to give me footnotes or endnotes. This is really scientific format. So you can cut and paste, but just give me the reference immediately following where you put your parentheses in your quote. Put it there right afterwards. So give me 3 advantages and 3 disadvantages. Now what I'm curious about is, as you look at advantages and the disadvantages, how do you vote? So you're going to give me a paragraph advantages, paragraph disadvantages, and then finally at the end just give me a couple sentences. What do you think? Should we be using biofuels or not? Just to let you know, there are some good advantages and there's some huge disadvantages. So I'm curious to see what you're going to find, and I'm curious to see if any of y'all will find biofuels' dirty little secret. And we'll talk about that in class as we review this paper. But yeah, there's something about making biofuels that they're just not telling you. And realize there's a lot of money in biofuels. So, always follow the money is what I always say, to see what's really going on. So that's, I will give you an announcement on Moodle to tell you when that little paper is due. It'll take you maybe an hour to do that. But this is a great way for me to help you get your grade up too. These tests are not the easiest things in the world. And so these papers I can be a little bit more lenient on, and to help you keep your grade up for this course. So, how do you vote papers? All right. Now, autotrophs and heterotrophs. Remember we talked about a little bit earlier, producers and consumers. This is just exactly the same thing again. This is producers and consumers. Autotrophs are the producers. Auto means self, troph means feeding. These are self-feeders. They get their energy directly from the sunlight. And so they're able to do that, obtain carbon from organic molecules. Plants and most other autotrophs make their own food by photosynthesis. Now, what is photosynthesis? Photo means light. Synthesis means to build. So it is building organic molecules with the energy of light. So animals and other heterotrophs, hetero means different, troph means feeding, so we have to get our food from somewhere else, somewhere different. We can't make it ourselves. So we get energy and carbon by breaking down the organic molecules assembled by the autotrophs. So the plants turn sunlight into sugar, and we eat either the plants or we're going to eat the consumers that ate the plants. So when we're eating a cow, that energy we're getting from the hamburger is actually energy that it got from eating the autotrophs. So remember, it starts with the sun, and all energy for life on Earth comes from these plants capturing sunlight in a chemical bond in sugar. So that's why this is so important. Remember, energy flows through all ecosystems on Earth. It begins when photosynthesizers intercept the energy from the sun. Photosynthetic organisms use pigments to capture the energy of sunlight and convert it to chemical energy. The chemical energy is stored in bonds, in sugars, carbohydrates, right? A monosaccharide, a simple carbohydrate. So photosynthesis, this will be a question on the test. I've got it highlighted here nice and blue for you. So the question will be, blank is the synthesis of organic molecules from inorganic molecules using the energy of light. And you just have to give me photosynthesis in the multiple choice. All right, so what is light? If you really want to irritate a physicist, ask them what light is, because they just really don't know. It has properties of energy. It travels in wavelengths. It has properties of matter. There's little pockets of energy called photons that acts like it is a particle of matter. So the consensus is still out. What exactly is light? Well, light does travel on wavelengths. The distance between the crests of two successive wavelengths of light tells us what the color is going to be and how much energy is there. The shorter wavelengths have greater energy. So here is our visible spectrum. In grade school, high school, you probably learned Roy G. Biv. You remember that? Roy, red, orange, yellow. G, green. Biv, blue, indigo, violet. Roy G. Biv. This is the visible spectrum of light. Now plants will absorb this visible spectrum, capture that energy that's in the photons of the wavelengths of light, and will use that energy to ultimately build sugars. And that's what this whole chapter is about. But you can see, here's our visible wavelengths of light. When we talk about wavelengths going everywhere from gamma rays to radio waves, which is the longest. But so here is purple. You can see the wavelengths are much closer. There's more energy there in purple, indigo, blue. And as we start going out toward yellow and red, you see they lengthen out. There's not as much energy. So ultraviolet light has a lot more energy than the infrared spectrum. All right, so different wavelengths form colors of the rainbow. We just saw that. Photosynthesis uses wavelengths between 380 and 750. So what is a pigment? It's an organic molecule that selectively absorbs specific wavelengths of light. Now these pigments, and I got some pictures of a few of them here, but they're pretty unusual. They have this molecule, but they also have this this chain of atoms sticking out of it that looks like it's an antenna, and that's basically what it is. It puts this little antenna up to catch a specific wavelength of light that comes through, and when that light comes through there, it's going to grab that photon and give it to an electron and get him all hyped up. And so that's what this is. So chlorophyll A is the most common photosynthetic. It absorbs violet and red lights and it appears green. Chlorophyll A is by far the greatest pigment in plants. That's why plants are green, chlorophyll A and chlorophyll B. They're green because of these two pigments. They absorb chlorophyll A, purple and red. So from now on, that should be your two favorite colors. You know, you're alive and have energy for your heart to beat and your muscles to move because of the wavelengths, purple and red. So there are other accessory pigments in the leaves. So, you know, they do absorb the rest of the wavelengths, but just, you know, by far the greatest is chlorophyll A with red and purple. So certain electrons in pigmented molecules absorb photons of light energy. It boosts the electrons up to the next highest level. Remember, we talked about the shell theory. So these electrons get that photon, they get all excited and they go up to the next level, but they quickly drop back down again. And when they drop back down again, so they'll grab that energy and then they pass it off, and we'll see that here in a minute. Energy is though captured and used in photosynthesis. So here is the main pigments that are in plants here. In our part of the world, there's chlorophyll A that makes plants look green. Here's a nice picture up here of the fall colors. You'll know why the leaves change colors in the fall. You know, we go from green leaves most of the year, and then just for a little bit they will turn red or yellow or orange. You'll know what's happening. Chlorophyll A and chlorophyll B break down quicker than all the other pigments. So right now the leaves look very green. Why? Because there's so much chlorophyll A and B that you can't see in any other pigments in there. But in the fall, they die off first. So now you start to see those pigments that is actually, they're working with chlorophyll. You just can't see them in the summer and spring. So now you're starting to see some of these others. Beta carotene, which gives that orange color. Lutein, which gives a nice yellow. Lycopene, which gives our plants that red color in the fall, the leaves. So all of these pigments are working together. And here, I don't know if you can see that real well. Yeah, it's showing up. Here's the molecule of chlorophyll A, and look, here's this string of little molecules added together here at the end of it to form like that little antenna that sticks up and will capture that wavelength of light, grab the photon and pass it on. Here's beta carotene. It actually has two larger molecules on either end with the sort of antenna-like thing there in the middle. It's not really an antenna. That's just the best way for me to understand what it's doing. So there's your take-home message, just a review of everything we just covered. Now, photosynthetic pigments work together to harvest light from different wavelengths. So as we said, chlorophyll A and B are by far the biggest, but the other ones are there for absorbing light energy too. Engelmann was a scientist who discovered, he was the first one to really prove that chlorophyll A is absorbing red and purple. Now how did he do that? He got a strand of Cladrophora, which is a green algae, and he placed it in a petri dish, and he also put in that petri dish bacteria that love oxygen. So when plants make photosynthesis, they release oxygen. So these bacteria are going to be swarming around where oxygen is being released. And so He put light on that through a prism which separated the Roy G. Biv. And here is what he did. So here's our strand of algae. He's got the light coming through, and look where the bacteria congregated. Much more down here in the purple section. Why was there much more? Because there's more energy here. So photosynthesis is taking place more quickly down here. But look where the other big cluster is. Down here at red. It's not as big as purple because there's not as much energy there. So this was Engelmann who proved that chlorophyll A will absorb the wavelengths of purple and red. All right. So photosynthetic organisms use the combination of pigments, though, so they're not just going to use those two little wavelengths. That'd be a lot of wasted light and a lot of wasted energy. So they have other pigments that will absorb the rest of the light spectrum. An absorption spectrum shows which wavelengths each pigment absorbs best. And so, we can see here's just different pigments, and it tells you the wavelengths that they are going to absorb. So here is chlorophyll A, and you see its peak is right here at purple, and down here is the other peak, which is at red, but you'll see the purple is greater than the red section. And here's beta carotene, which is orange, and then there is phycoerythroblin, man, that's a word. But you see that they use all the different wavelengths with all the different pigments that are inside of the leaf. So again, there's our take-home message, just a review of what we just talked about. So an overview of photosynthesis. In plants and other photosynthetic eukaryotes, photosynthesis occurs in the chloroplast. Photosynthesis also occurs in two different stages. There is a light-dependent stage. When I was growing up, we learned there's a light stage and a dark stage, but that makes you think that actually the dark stage happens in night, the light stage happens during the day. No, actually, it should be a light independent. It goes on all the time. That's where sugars are built. But it doesn't need light. So it can happen at night. It happens all the time. So they changed those two stages. Also, in the light-dependent reaction, so we'll start off with that. The first stage is the light-dependent. There's actually two pathways. The first is the non-cyclic pathway. Now, I'm going to be going through these next couple slides, and just realize it's not going to make much sense to you right now, because to me, a picture is worth more than 1,000 words, and so we need to see these pictures to really understand. So I'm going to be going through these slides, but it's going to make a lot more sense when we see the pictures of how this takes place. So in the non-cyclic pathway, light energy is transferred to two coenzymes. ATP, remember that is our energy unit of the cell. It was a DP, it had two phosphates, but through this process we're going to add another phosphate to it, and in that bond holding on that phosphate group is going to be the stored energy that your body uses. But also it's going to form NADPH from NADP. It's going to add a hydrogen to NADP. So that is the non-cyclic pathway. It has two products, ATP and NADPH. But in that also water molecules are split. It releases oxygen. So remember, in photosynthesis, oxygen is released. The light-dependent reaction occurs in the thylakoid membrane. And that's inside the chloroplast. It is a folded membrane that makes up the thylakoids. And the thylakoids have these clusters of light-harvesting pigments that absorb photons of different energies and convert light energy into chemical energy. Now that's a lot to talk about right there, but it's going to make a lot more sense once we get to these pictures. All right. Energy in ATP and NADPH drives the synthesis of glucose and other carbohydrates. So now we're in the light independent. So remember, in the light dependent reaction, uses sunlight to just make two things, ATP and NADPH. Then the light independent reaction uses those two products of the first part to form glucose and other simple carbohydrates. from carbon dioxide and water. It occurs in the stroma of the chloroplast. The stroma is that semi-fluid matrix surrounded by the two outer membranes of the chloroplast. Sugars are built in the stroma. That's a test question. Where are sugars built in the stroma? Where does the light-dependent reaction take place? In the thylakoid membranes. All right, so here's our formula for photosynthesis. This is just a very nice, easy formula. Move this out just a little bit. So 6 carbon dioxide plus 6 water with light energy yields, this is glucose, C6H12O6, this is a sugar, and oxygen. So carbon dioxide and water and light energy work together, and our product is sugar and oxygen. So the chloroplast, that's that organelle that specializes in photosynthesis in plants and many protists. That thylakoid membrane, so this is the same information again, but this is where the first stage of photosynthesis takes place. Can't wait till we'll get to the picture here just in a second. The stroma, there's the definition of the stroma again. That's the second stage. Sugars are built in the stroma. Know that. Sugars are built in the stroma of the chloroplast. So here finally is our chloroplast. If you can see up there at the top, that's actually a slide. You can see the cells of a plant and you see the chloroplast in here. This is a chloroplast. This is the thylakoid. They look like pancakes stacked up on each other. This, though, the thylakoid membrane is one continuous chamber. So you can start over here and go all through, going back and forth, and it's like a maze. It never ends. So it's one continuous chamber inside, and you can see here, this is what it looks like inside. So this is where chlorophyll is, on the thylakoids. This is where the other pigments are, on the thylakoid. This is what absorbs the light, grabs the energy of light, and makes ATP and NADPH. Now, the ATP and NADPH is then going to be out here in the stroma, which is this fluidy matrix around it. And then the second part of photosynthesis, the light-independent reaction, takes place here, where the ATP and the NADPH, the energy stored in there, is used to build sugars. All right, so there's our take-home message. So there's chloroplast again, there's the stroma again, there's this statement again. It's repeating itself, I know. Remember, learning comes through repetition. You need to keep going over these things. Where does the light-dependent reaction take place? In the thylakoids. Where are sugars built? In the stroma. Again, now, we got some new information here. On the thylakoid membranes, we have two photosystems. We have photosystem 1 and photosystem 2. Why are they named that? Photosystem 1 was discovered first, and photosystem 2 was discovered second, so they're 1 and 2. But there's groups of molecules in these photosystems that work to begin the reactions of photosynthesis. And they start the process of converting light energy into chemical energy. The photosystems don't do that, but they start the process. So there's our light-dependent reaction. What's being done here? Light is transferred to ATP and NADPH. Water molecules are split, releasing oxygen. Energy in the light-independent reaction, the ATP and NADPH, that energy is used to build glucose molecules and other simple sugars from carbon dioxide and water. So that is just the overview of photosynthesis. There's our set formula again. Know that formula for the test. I'm not going to ask you the numbers, but just know you need water and carbon dioxide. That's your reactants. Then with the light of energy, that's our catalyst. Look which way the arrow's going. So this is the products over here. Oxygen and a sugar. All right. This is just a breakdown of a plant cell. We're going to look at these more in depth as we go through the chapters on plants. But notice here's the top of the leaf. The cells that have the chloroplasts are packed more tightly near the top. Why? Because that's where the sunlight's hitting. That's where the most sun is. Down here in the spongy mesophyll, it's not nearly tightly packed, but this is where, here's an opening here, the stroma, where the carbon dioxide is able to come in so that sugars can be built, and the oxygen released is able to go out. So this is the leaf where photosynthesis takes place. Again, there's our chloroplast with the thylakoid membranes and with the stroma, and this is where photosynthesis begins. This is just a breakdown again of the light-dependent and light-independent. You can see this is built as a wheel. This is actually a cyclic pattern that builds sugars. But what does the light-dependent reaction do? Sunlight is used to make NADPH and ATP. The light-independent reaction then uses those two coenzymes to form sugar, and now these are recycled back over to the light-dependent reaction so that they can pick up a phosphorus and be ATP again, and pick up another hydrogen ion to be NADPH. And so these things are reused over and over and over. Now, can't wait to get to the real picture here. All right, the light-dependent reaction. This is the first stage. Light energy drives electrons out of photosystems. The electrons may be used in a non-cyclic or cyclic pattern of ATP formation. So the photons boost the electrons in pigments to the next energy level. The light harvesting complexes absorb that energy, and the electrons are released from special pairs of chlorophyll A molecules in photosystems. So here is a close-up look at the outside of a thylakoid membrane. Notice the light harvesting systems here, and here are the photosystems. Now what happens? This is in the leaf, it's out there so the sunlight can hit it. So the sun hits it, and what happens? It's got its antenna up, it grabs the photon out of the sunlight, and it gives it to an electron. That electron just gets really excited, gets hyped up, but he quickly will jump back down. He will release that photon, and that photon is passed back and forth through these light harvesting complexes. The photosystems, think about them as a basket. So they're playing basketball. They got this photon, it's like a hot potato. They're passing it back and forth until there's an opening in the photosystem. They shoot, they score. The photon goes down through the photosystem, and there is an electron there. So this is where all the chlorophylls are, but there's an electron there that is going to grab that photon, get all excited. Remember we talked about this, that when they absorb that extra energy, it's like a person who just drank 5 Monster drinks. They got all this energy and they're just bouncing off the walls. And so what's going to happen now, let's get past this, is that he will pass that along. I don't want to skip ahead too far here without giving you the pertinent information. All right, we've already covered that. Now there's something you need to understand here that's going to take place, and this is really important. Photolysis. Photo again means light. Lysis means to break apart. So think about that. The chlorophyll A has this electron. Actually, they have a pair of electrons. And one of those electrons is going to grab that photon, that burst of energy, and he's going to get all excited, and he's going to go down through that photosystem. Now suddenly, chlorophyll doesn't have 2 electrons anymore. It can't absorb any more photons of light, so it's got to constantly be replenishing those electrons so that new electrons will be there to grab the next photon of light, and they can go and start using that energy to make sugars. So photolysis is the process by which light energy breaks down a molecule such as water. That's what's going to happen. Why is water critical for photosynthesis? Water is there, and the energy of the light is not just going to be absorbed by that electron, it's going to be used to rip a water molecule apart. So remember what water is. It's 2 hydrogens, 1 oxygen. It's going to rip the hydrogens off. They're now hydrogen ions. And it's going to release the oxygen outside. So this is the first time that oxygen is being released in photosynthesis. Where is it coming from? From the water molecule. Now why is it doing this? So that there will be electrons in chlorophyll A and B and all the other pigments, electrons to absorb those photons as they come through. So that's photolysis. And then, once that electron gets all that energy, remember he goes through these electron transfer chains. Remember when we looked at that, it's like a little stairway. So he gets all that energy from like drinking 5 Monster drinks, and so he's going to run down the stairs to work off that energy. And as he goes down each step, he's going to be releasing just a little bit of energy. And the plant cells are going to use that little bit of energy he releases for an enzyme to grab a hydrogen ion outside the thylakoid and bring it inside. Remember, active transport. This is active transport, but it's using the energy of the sun here from an excited electron to grab hydrogen ions and bring them in. All right. So light energy is converted to chemical energy. Yeah, we've talked about that. Entry of electrons from a photosystem to the electron transfer chains is the first step of the light-dependent reaction. Then the second part. ATP forms in the stroma. Electron energy is used to build up a hydrogen ion gradient across the membrane. Hydrogen ions flow through ATP synthase, which attaches a phosphate group to ADP. That don't make any sense right now, right? Let's get to this. This is what I've been waiting for. I hope you can see this well. This is important. You have a PowerPoint of this also. This is the thylakoid membrane. So here's the inside of the thylakoid membrane, and here's the outside. Notice there's a lipid bilayer, just like every other organelle and every other cell. That lipid bilayer allows things to come in selectively. So water can usually flow in and out, but it has to actively pump in larger molecules or certain molecules. So here is the thylakoid membrane. Here is photosystem 2. We're going to start with photosystem 2 because it is more important. All right, so here's light energy. Hits photosystem 2. There is our chlorophyll. All right, absorbs that photon, gets really excited, and now that electron is going to come here, and here is the staircase. This is the electron transfer system. Now, suddenly, chlorophyll had 2 electrons. Now it's only got 1. It can't do the job. So here's our water molecule. Here is photolysis. It rips off the hydrogen ions and it releases the oxygen. The oxygen comes out, the hydrogen ions are inside. Notice what's happened here. Light energy is increasing the concentration of hydrogen ions inside. So water has to keep coming in so this can keep functioning, but hydrogen ions are starting to build up. Let's see what photosystem 2 does. So now, the electron's got the photon, he's got all that energy, he goes down the stairway, and each time he goes down the step, this enzyme here will use that energy that he releases to grab a hydrogen ion from outside and pull it inside. You see what sunlight is doing? The only thing that sunlight is doing here in the thylakoids, it's pulling in hydrogen ions. It's ripping off hydrogens from water, so we got hydrogen ions coming this way. And as it goes down the electron transfer chain, it's bringing in hydrogen ions from the outside. That's what sunlight is doing. That's it, pulling in hydrogen ions. Now, we've got a whole bunch of hydrogen ions on the inside, very little on the outside. Remember last chapter we talked about diffusion, the movement of molecules through their concentration gradient. So what are these hydrogen ions wanting to do? They want to, there's a bunch of them in here, very few out here. They want to move this way, but they can't. So they keep hitting this plasma membrane. We got to get out and even everything out, but we can't. So they're looking for a way out. There is a way out. There's only one way out, but there is a way out. All the way down here at the end, here is a big honking protein. This is an enzyme. The name of this enzyme is ATP synthase. I'm so glad they named it that because that's exactly what it does. It builds ATPs. Now what happens is this is the only place that hydrogen ions can go out. So as it goes out, it energizes this enzyme to grab a phosphorus and add it to an ADP. So there's a bunch of ADP out here. As the hydrogen comes out, it gives it the energy to grab a phosphorus and attach it to the ADP to make ATP. Remember, ATP is the energy unit of the cell. All right, so what has photosystem 2 done? Light hits photosystem 2. The electrons in the chlorophyll get really excited and go down the electron transfer chain. To replace that electron, Water then goes through photolysis using the energy of the sun to break down a molecule. Oxygen is released, hydrogen ions stay in the dipole. As this electron now goes from photosystem 2, goes down its electron transfer chain, down its steps. Each step it goes down, it allows another hydrogen ion to be pulled in. So what is light from photosystem 2 doing? Pulling in hydrogen ions. Then those hydrogen ions, by diffusion, will naturally flow through the only way out, which is an enzyme called ATP synthase. And every time it goes through, I like to think of it as the hydrogen ion goes through, it causes that enzyme to turn. And as it turns, it takes a phosphorus and adds it to an ADP. So what's happening so far is we're making ATP through a hydrogen ion gradient. Now, we still got another photosystem. Here's photosystem 1. All right, so photosystem 2, the electron grabs the energy, goes down the steps, pull in hydrogen ions. Here is our electron at the end of that. He's at the bottom of the steps and he's like, oh, man, I just crashed. I don't have any energy. I don't have any purpose in life. Oh, he's suddenly Eeyore. And everything's negative, right? I don't have any energy. There's nothing I can do. And he is negative because he's an electron. So he is now going to go to photosystem 1 because in photosystem 1, he's going to go into a pigment. And so now he's there, all right, I'm going to get another burst of energy. I just know it. And sure enough, light hits him and he gets that burst of energy. And now he's got, he's hyped up again and he's going to run down the stairs again. And as he goes down the steps of the electron transfer system, this time, instead of pulling in hydrogen ions to make ATP, there's NADP out here in the stroma. And that energy is going to be used to grab a hydrogen ion that's probably been released from the cell as it goes through to make ATP. So there's all these hydrogen ions out here. It's going to take one of those hydrogen ions and add it to an NADP to make NADPH, another coenzyme that holds energy. So that is the light-dependent reaction. This is the non-cyclic pathway of the light-dependent reaction. But this is the most important. This is the beginning of photosynthesis. So it's a beautiful process, amazing process, that all this energy is made by just pumping hydrogen ions across a gradient and allowing them to go out through an enzyme, and then using another energy gradient to take a hydrogen ion and put it on an NADP. Now, that's the non-cyclic pathway. As the thylakoid makes ATP and NADPH, it can make NADPH much quicker than it can ATP. So, especially when it's really sunny outside, NADPH can outmatch ATP. And so we're going to see as we go through the next stage, it needs both. So what it's going to do is it's going to shut down NADPH production and focus only on ATP production. And you would think then it's just going to shut down photosystem 1 and let photosystem 2 just keep going. That's not what the plant does. Why? I don't know. What it's going to do is going to shut down photosystem 2. So what's going to happen here is when photosystem 1, the electron goes down at the end, and what he's going to do, he's going to recycle. So the same electron is just going to be going around and around and around. It's like little kids at the amusement park. They ride the roller coaster, and woo, that was great. And so they run around as soon as they get off to get back in line again. That's the cyclic system. The same electron going around and around and around. But this time, this energy is going to be used not with this enzyme to form NADPH. That energy is going to be used with another one of these enzymes to pull in hydrogen ions. So now only ATP will be made. So photosystem 1 in the cyclic pathway will stop making NADPH and start pumping in hydrogen ions. Photosystem 2 shut down. The break apart of water shut down. Everything shut down except for these electrons just going around in circles here and making ATP by pumping in hydrogen ions. So that is the cyclic system. And you know, the plant, you know, keeps turning this on and off. Most of the time, it is the non-cyclic pathway, photosystem 2 and photosystem 1. It's just when the levels of NADPH start to get too high, then they're going to be wasted because you have to have ATP with it. That it's going to shut down the production of NADPH and just focus with photosystem 1 to make ATP. All right, so when NADPH accumulates in the stroma, the non-cyclic pathway stalls. The cyclic pathway runs in type 1 photosystem to make ATP. We just talked about that. Electrons are cycled back to photosystem 1, and NADPH does not form. All right, so that is the light-dependent reaction. Look at it in your book. If you got a book, you just go over that again and again and again. Watch this little section of the video again and again and again, because there's going to be a bunch of test questions from this picture. What is photolysis? Where does the electrons come from that supply the electron pairs in chlorophyll for photosystems? For photosystem 2? Well, it comes from water. What does the energy from the photon from photosystem 2, what does that use to pull inside of the thylakoid? It's hydrogen ions. In photosystem 2, light energy is captured to form what? NADPH. And in the light-dependent reaction, the hydrogen ion concentration inside leaves through what that forms ATP, and that's ATP synthase. So there's going to be a lot of questions from this one little slide here, but you know, it's all these other slides that have been pouring into this too. But this is the miracle of the light-dependent reaction. Taking sunlight and capturing that energy into these two coenzymes, NADPH and ATP. All right, let's get back to where we were. All right, that was the. All right, energy flow in the light-dependent reaction is an example of how organisms harvest energy from the environment. So this is how the producers get their energy. All right, here's a term you're going to need to know for the test: photophosphorylation. Photo means light, phosphorylation means adding of phosphorus. So remember, in the light-dependent reaction that brings in the hydrogen ions, they leave to the ATP synthase to do what? To add a phosphorus to ADP. So that is photophosphorylation, using the light of the energy of light to add phosphate groups on. So in the non-cyclic photophosphorylations, electrons move from water to photosystem 2 to photosystem 1 to NADPH. In the cyclic phosphorylation, electrons cycle within photosystem 1. So we talked about that. So, you know, I'm not going to ask you questions on this. To me, this is even more confusing. I just like that last picture that we spent time on, talking about what the light-dependent reaction is. All right, there's the cyclic pathway, only photosystem 1. All right, now, that's the first part. So what is the product so far of photosynthesis? ATP, NADPH, that's it. So now, what's the plant going to do with that? Because ATP and NADPH is not a great way to store energy. It's for quick energy, but not for storing energy. So the plant is not going to use that ATP and NADPH in the chloroplast for energy. It's going to work now to fix it into a fixable energy, and it's going to use that, the energy in ATP and NADPH, to make sugars. Realize something, sugars are extremely stable. When they unearthed King Tut's tomb, they discovered a pot of honey in his tomb that was still just as good as the day that it was put in there. Sugars will not break down naturally. They are a very stable molecule with that energy trapped in those bonds. Both of my sons like soccer, and you know, I've always thought soccer was kind of boring because I hate watching a game and it ends up 0-0. As boring as I think soccer is, I always give this example. If it wasn't for us breaking down sugars that that man, the game would last forever. It would take us over a year for sugars to break down naturally to get enough energy to kick a soccer ball even once. And so we're going to talk about that in the next chapter, breaking apart sugars. But man, these compounds are stable once they're made into a sugar. So that's why plants are going to go through this cycle. The Calvin-Benson cycle. Enzyme-mediated reactions that build sugars in the stroma of the chloroplast. So know that term for the test. The Calvin-Benson cycle is the second stage of photosynthesis. It is the light-independent reaction, but we call it the Calvin-Benson cycle because of the two men who discovered it. All right, carbon fixation. This is critical. It's the extraction of carbon atoms from inorganic sources and incorporating them into an organic molecule. Remember we talked about as we started this, where does all the weight in the trees come from? Well, it comes from the air, carbon fixation. It is almost weightless in the air, but when you start adding them together to form bigger molecules, they start to have weight, and so we're fixing carbon from air into something that is solid. Carbon fixation builds glucose from carbon dioxide. It uses bond energies from the molecules formed in the light-dependent reaction. So here's our two products of the light-dependent reaction, ATP and NADPH. It's going to use these two to make a sugar. The enzyme rubisco. Know that enzyme for the test. This is the most, it's the most plentiful enzyme on the planet, probably the most important enzyme on the planet. It is going to grab a carbon dioxide, break the carbon off, and fix it to a carbon chain. It is the process of carbon fixation. I'm not going to be asking you to test these groupies and PGAs. To me, the PGA is the Professional Golf Association. I don't know what that has to do with making sugars. I'm not going to ask you all these intermediates. I just want you to know the Calvin-Benson cycle takes the ATP and NADPH and builds a sugar. Rubisco takes the carbon off of carbon dioxide and fixes it onto a carbon chain. Rubisco is then regenerated. It keeps doing this again and again and again. All right, again, we've already covered this. Here is the ATP and NADPH from the light-dependent reaction. Now we've got carbon dioxide coming into play, but here is the light-independent reaction, the Calvin-Benson cycle. What will be the products? Main thing is sugar, and since we're using this energy from ATP, we're going to end up with ADP, and since we're using the energy from the hydrogen ion attached to NADP, we're going to take that hydrogen off, and it's going to be the product here. So that just makes sense. So here is the Calvin-Benson cycle. Let's see here. I think I got a better close-up here of it that you'll be able to see a little bit better. This is how sugars are built. This takes place in the stroma. So there's the picture up there, it's in the stroma. So the ATP and NADPH is coming out of the thylakoids from the light-dependent reaction. All right, so here is this sugar. Let's count the carbons. 1, 2, 3, 4, 5. A 5-carbon carbohydrate called RUBP. There's 6 of them. Look here, we have 6 carbon dioxide molecules coming in. So this is a wheel. It doesn't really look like a wheel, but this is showing us that this is a cycle. So 6 carbon dioxides. Right here is rubisco. Rubisco is going to take these 6 carbon dioxides. There will be 6 different rubisco molecules here. They will take the carbon off, release the oxygen, so oxygen goes out, and they're going to add a carbon to each one of these 6 RUBPs. Now there was 5 carbons there. When you add a carbon, how many carbons do we have then? 6, right? Now remember, glucose is a 6-carbon sugar, but this is not glucose yet. What's going to happen is, as soon as this gets to be a 6-carbon, RUBP will then split in 2 to form 12 PGAs. This is a 3-carbon chain. Now, here is where, look, here's ATP, and there is NADPH. So look, we actually need more ATP than we do NADPH. That's why we will shut down photosystem 1, forming NADPH to make more ATP. We just need more ATP. All right. So we take the energy from ATPs, look, there's 12 of them here, and guess what those do? It's ATP, so it's got an extra phosphorus. It's going to add a phosphorus to the PGAs. Then the NADPs, H's, is going to use that and attach a hydrogen to it also. So what we come up with at the end of this is 12 PGALs, I call them PGALs. And so there's 12 of those. There's 12 3-carbon chains. Now, here is where sugar production takes place. 2 of these will be put together to form 1 glucose or another sugar. I mean, there's other sugars other than glucose. Glucose is the main one that plants make, but there's other simple sugars. Now, since we use 2 of these P-gals, what's left? 10. You know, that's a simple subtraction, right? Even I can do that. Now, what's going to happen? Well, they're going to be reorganized into RUBP again. So now we will start this whole cycle. And it uses 6 ATP to reform those into RUBPs. So this is the cycle. Now, what do you need to know about the Calvin-Benson cycle? First of all, know what's the Calvin-Benson cycle. This is where sugars are made in the stroma. It uses the energy of ATP and NADPH to form a 6-carbon glucose molecule. Where does the carbon come from for that glucose is made out of? It comes from carbon dioxide. Know that rubisco is the enzyme that removes the carbon from carbon dioxide, attaches it to that, and then those carbons will be reassembled down here to form the glucose molecule. The rest of it will be recycled so that RUBP is made again, so that it can take new carbon dioxides, go through this cycle again, and just keep making sugar. So this thing is going to keep pumping out sugars. And realize this doesn't just happen one place in the stroma. This happens thousands upon thousands of places in the stroma of a single chloroplast. So this thing is going on throughout the chloroplast and the stroma. All right, that's about all you need to know for the light independent reaction, or the Calvin-Benson cycle. Here's just another view of it. There's our RUBPs. There is our carbon dioxide. Here's rubisco. Forms 12 PGAs. So we go from a 5 to a 6, and then breaks in half to go to the 3s. ATP and NADPH to form the 12 PGALs. And then 2 of those get together to form glucose. 10 of them are left. They are restructured to form the RUBP. So this whole thing starts again. But notice here's the thylakoid membranes. This is in the stroma. It's not inside the thylakoids. That's the light-dependent reaction that's making the ATP and the NADPH. This is happening in the stroma. All right, I know that's a lot of information, but realize this is the basis of life. How does energy get captured from the sun so that things can live on this planet? It's those 2 reactions. So there is the light independent and the light-dependent phases of photosynthesis. Now, that's a lot. I'm going to go over this material right here with you. I'm not going to ask it on the test. What this next part is about is because of different environmental conditions around the world, that some plants have to change slightly the way that they make these sugars, like in deserts where there's not much water, and it is so hot that when they open the stroma and the leaves to allow carbon dioxide in, that the water there evaporates out. So they have to close the stroma, and so they don't have an influx of carbon dioxide. How do they get around that to make sugar? So that's what these different types of plants are. We have C3 plants, C4 plants, CAM plants. Just go through this real quickly. Again, this is just for your edification. I'm not going to ask you this on the test. I think if you've got just the basics of photosynthesis down, that's enough for an entry-level biology class. I just want you to know that, yeah, there's conditions where plants have to change what you just learned a little bit so that they can keep making sugars in any conditions. So, what are the stomata? Small openings through the waxy cuticle covering the epidermal surfaces of leaves and green stems. It allows carbon dioxide in and oxygen out. They close on dry days to minimize water loss, but also really when they close, new carbon dioxide can't come in and the bad oxygen for the plant can't come out. Closing, however, prevents gas exchange as well, so there's a problem on dry, hot days. When the stomata closed, oxygen levels rise, carbon dioxide levels decline. This reduces the efficiency of sugar production in the Calvin-Benson cycle, right? Because it's got to keep making that ATP and NADPH, and it needs water at all times, and it needs carbon dioxide to make the sugar. So both cycles are dependent on those stomata being open. So what is photorespiration? At high oxygen levels, when the stomata close and it can't get rid of the oxygen, rubisco will then attach an oxygen instead of a carbon. It is an inefficient way to produce a sugar. It's a different sugar. It's not an efficient way to do it. It requires more ATP and NADPH. But when there is conditions where the plant can't do anything else, or else it's going to dry up, then it will close the stomata and it will go that route. CO2 is produced rather than fixed. So instead of releasing oxygen, it's going to start releasing carbon dioxide, which is not optimal by any stretch. So C3 plants are plants that use only the Calvin-Benson cycle to fix carbon. Forms a 3-carbon PGA in the mesophyll cells. It's used by most plants. So that's basically what we just learned, is C3 plants. It's inefficient in dry weather when the stomata are closed. So barley is an example. All right, so I'm not going to be asking about this. I just want you to know they're there. C4 plants that have an additional set of reactions for sugar production on dry days when the stomata are closed. It compensates for the inefficiency of rubisco. If rubisco is going to start attaching oxygen instead of carbon dioxide. So it forms a 4-carbon oxoacetate in mesophyll cells, then bundle sheath cells make sugars. Example, corn, switchgrass, and bamboo. I'm not going to ask you this. I mean, that's a lot of information to try to process how it does this. I don't have the pictures for how it does it, the really good pictures. I'm not going to make you do that. This is what I have, and that's not a great picture, but here's the Calvin-Benson cycle down here, an adapted Calvin-Benson cycle. All right, TAM plants. Plants with an alternate carbon-fixing pathway that allows them to conserve water in climates where days are hot. Desert plants, TAM plants, cacti, things like that. Forms a 4-carbon oxaloacetate at night, which is later broken down to carbon dioxide for sugar production. So it's making its own carbon dioxide, which is pretty neat, really, that these plants can do that. But cactuses, succulents do this sort of thing. So there is the diagram for that. All right, so biofuels revisited. I will give you more information on biofuels again. I'm not going to ask you these terms, but just know that I'll attach on your announcements for the class. I'll send out an email to you also to make sure you get it. How to write this paper. Again, 3 positives, 3 negatives. How do you vote? But we talked about the effects on atmospheric oxygen. We'll cover this more in the next chapter. I'm not going to worry about that right now. That's just, you've got enough for this chapter already to cycle in. All right. Fossil fuel emissions, they're small. We'll cover that in the last chapter we do this semester. Biofuels, again, are renewable. The carbon in plants comes from atmospheric carbon dioxide. But just realize There are some positives. It does burn cleaner. There's some negatives. And I want to see what negatives you come up with. There's a bunch. To me, one of the most blatant is we're taking food and making gas out of it. It's moonshine, ethanol moonshine, and then putting that in gas. When there's people starving all over the world, and we're turning food into gasoline. Only in America, I think, is this something we think, hey, that's a really good idea. And this ethanol is really hard on our engines. The engines break down much quicker with ethanol present. So our engines would last much longer without ethanol. But I'm curious to see if you're going to find this dirty little secret. I think Stanford was the first one to really publish this out. That what was the purpose of making biofuels? To clean up the environment. See if you can find a source that says, actually by making the biofuels, we're actually polluting the environment more. So as of right now, we're going to find ways in the future to make these biofuels more efficiently, but just see if you can find that. So that's chapter 6, photosynthesis, and get ready for chapter 7. So we've made sugars in chapter 6. Chapter 7, we're going to be breaking apart sugars to get that energy back out. So looking forward to going over that with you. We'll see you soon.